Students, now let us see some multiple choice questions from the chapter Cell the Unit of Life. So there will be some kind of tricky questions which will be asked in the NEET examination. You should read the question once or twice in order to understand the question, and then you should start to find the answer. So in a hurry, you should never find the answer. You will end up making a mistake. So now let's see how to solve some tricky questions that can be asked in the NEET examination. The first question, the ratio of percentage of lipids and proteins in erythrocytes of membrane. So this is a direct question. The information is directly given in the NCERT lines. So in plasma membrane of erythrocyte, the lipid content will be considerably less and protein content will be more. That will be 52%. Protein will be there and lipid percent will be only just 40%. So lipids will be 40, proteins will be 52. D is the correct option. Okay, it is not 52 is to 40. Okay. Next, let us go for second question. Find the mismatch. Which is the mismatch here? 4 options are given. Rough endoplasmic reticulum, ATS ribosomes. Or here will have ATS ribosomes. This is correct match only. Middle lamella contains calcium pectate. This is also correct match. Leucoplast starch synthesis. Look here students, leucoplast does not carry out starch synthesis. Only chloroplast can synthesize starch. There will be leucoplasts called amyloplasts which can store starch but not synthesize starch. So C is the mismatch. Histone proteins, basic protein. Histone proteins are basic in nature. This is also correct match. So correct option is C. Next, let us go for third question. Look here. The longest portion of flagellum is. So in a flagellum structure of prokaryotic cell, which is the longest part students? Filament. This part. This is the filament. Okay. So direct question, filament is the longest part, not hook, not basal body, not the centriole. Okay. Next, fourth question, the extensions of plasma membrane which form mesosome are not in the form of. Students, below the cell wall you can see the plasma membrane form extensions like this, which is called as mesosome. The mesosome occurs not in the form of, okay, the mesosomes occur in the form of vesicles, tubules and lamellae, but not in the form of a network. So C is the correct option. Next, fifth question. Which is the site of formation of glycoprotein? Glycoprotein is formed in which of these cell organelles? Is it in RER? No. RER will synthesize only proteins, not glycoproteins. SER? No. They will synthesize lipids. Ribosomes? No. Ribosomes will synthesize only the proteins. Golgi complex? Yes. Golgi complex is involved in synthesis of glycoproteins, in the formation of glycoproteins and glycolipids also. Okay. Next, sixth question. Secondary cell wall is formed towards. In which cells we can see secondary cell wall students? In the plants. Okay. So in the plants, in the plant cell, look here. In the young plant cells, we can see already formed cell wall which is called as primary cell wall. This is called primary cell wall. Later, during maturation process, what happens? Second cell wall is formed towards inside, facing the plasma membrane. This is called secondary cell wall. This is called secondary cell wall. Okay. So secondary cell wall is formed towards membrane. Towards membrane means towards inside. Plasma membrane will be here. Okay. So towards the membrane, secondary cell wall will be formed. A is the correct option. It is not towards middle lamella because middle lamella will be present here between the two cells, adjacent cells. Middle lamella will be here, not towards outside. So middle lamella will not come. Mesosome is not found in plant cell. Okay, they just gave to confuse you. So mesosome is not the answer. Glycocalyx, this is also seen in the prokaryotic cell. This is not the answer. The correct option is membrane, towards the membrane. So like this tricky questions can be asked in the NEET exam. You should read the question twice or thrice. Okay, then carefully you should identify the answer by logical thinking. So this is how you have to solve the question, students. Students, now we will discuss few extra points which are out of NCERT. If you know these points, you will have always an upper hand over other students in the NEET competition. Now let's see what is primary cell wall and what is secondary cell wall in plant cells. So here, primary cell wall and secondary cell wall, they are found in plant cells. So when they are formed and towards which side they are formed, we will discuss now. Look here, this is a plant cell. This is a plant cell, a typical plant cell. Okay, this is the cell wall. The first formed cell wall in a young plant cell, always it is called as the primary cell wall. Primary cell wall, okay, and the plasma membrane will be towards inside. This is the plasma membrane. This is the plasma membrane. Okay, so the primary cell wall, it will be made up of material called cellulose. Cellulose. Cellulose is elastic in nature. Elastic. It is having elastic nature. So because of this elastic nature, the primary cell wall will allow the growth of young cell. Okay, so the primary cell wall, it allows growth. It allows the growth of young plant cell. Okay, eventually when cell it becomes matured, then what happens? Another cell wall is formed over the existing primary cell wall towards the membrane. So this newly formed cell wall, it is called secondary cell wall. Secondary cell wall. So the secondary cell wall, it is made up of variety of cell wall materials like lignin. In some cells we can also see suberin. So this type of materials will form the secondary cell wall, and always the secondary cell wall will be rigid. It will be rigid material. So once the secondary cell wall is laid down, then there will be no scope for the growth. The shape of the cell will become permanent. Okay, so this is about primary cell wall and secondary cell wall. I repeat, students, primary cell wall is present in the young plant cell, and it is the first formed cell wall. It is always made up of cellulose and it is elastic. It allows the growth of the young plant cell. So when cell matures, then what happens? Another cell wall is formed over the existing primary cell wall towards the membrane. So look here, secondary cell wall is formed towards membrane, towards inside. Okay, and secondary cell wall is composed of materials like lignin and suberin, and it is always rigid. It does not show any scope for the growth of the plant cell. So this is about primary and secondary cell wall. Okay, so now let us discuss about another concept of gram positive and gram negative bacteria. You can copy down if you want. Okay, students, now we will discuss the concept of gram staining, which was given by a scientist called Christian Gram. Gram staining. This procedure was given by a scientist called Christian Gram. By using the procedure called gram staining, we can stain the bacteria into two different colors, purple color and pinkish color. Okay, those bacteria which stain purple are called gram positive bacteria. Those bacteria which stain purple will be called as gram positive bacteria, and those bacteria which stain pink will be called as gram-negative bacteria. Okay, so this staining procedure is based upon the chemical nature of cell wall. So what differences we can see in the cell walls of gram-positive bacteria and gram-negative bacteria we will see here. So gram-positive bacteria will have more peptidoglycan component, whereas gram-negative bacteria will have less peptidoglycan, less amount of peptidoglycan in the cell wall. Okay, so next, in gram-positive bacteria there will be teichoic acid will be present, whereas in gram-negative bacteria teichoic acid will be simply absent. Okay, next, in gram-positive bacteria there will be lipopolysaccharides absent. Lipopolysaccharides are absent, whereas in gram-negative bacteria lipopolysaccharides will be present. Okay, next, cell wall is thick in gram-positive bacteria, whereas in gram-negative bacteria cell wall is thin. Okay, so I can just draw a small diagram for identification. Gram-positive bacteria will have thick cell wall like this. Okay, gram-negative bacteria, it will always have a thin cell wall like this. Okay, so because of the chemical nature of the cell walls, what happen, the gram-positive bacteria will take a purple color stain and they appear purple, and because of the chemical nature of cell wall in gram-negative bacteria, they take a pink stain and they appear pink in color under the microscope. So this whole procedure was developed by scientist Christian Gram. Understood, students? Copy down this. We'll meet in the next class and we will continue. So thank you. All the very best. Till then, keep reading and be happy.